Let's say you want to image through your finger. What color of light will you use? Well, we can do a very simple experiment. We can put a white light just behind our finger. We can see only red light goes through. It means that blue light and green light with a shorter wavelength get absorbed or scattered by our finger. But red light with a longer wavelength has a better optical penetration through our finger. So the answer is simple. If you want to image through your finger, you use red light at a longer wavelength. In neuroscience, instead of the finger, we want to image through the brain, which is essentially like a piece of tofu. It is highly scattered. And what's even more challenging is that we want to have cellular resolution and image as wide and as deep as possible, so that we can capture as many neurons as possible to know how they function together. So how are we going to do that? Hi everyone, I'm Aaron, a PhD student from Professor Christie's group in Cornell University. The first question will be, how can we go deep? Previously, people have used two-photon microscope to image deep into scattering tissue. In one-photon microscopy, you can see that above and below the focus, there are a lot of fluorescence. And these fluorescence will contribute to the background in the images and eventually degrade the contrast of the image. However, for two-photon microscopy, you can see the focus is really sharp on the focal plane. There's no fluorescence above or below the focus that may eventually degrade the contrast. However, even for two-photon microscopy, there's still an imaging depth limit for a densely labeled brain. You can see this image series here at a 600 micrometer depth. You can see that the image's contrast start to degrade, and we cannot see the neuron very clearly. So now we may ask, how can we image deeper than 600 micron? Well, if you remember the example I gave in the very beginning, if we want to image our finger, we use red light with a longer wavelength. So the logic follows here. If we want to image deeper beyond 600 micron, we can shift to a longer wavelength. This graph here shows the benefit of shifting into a longer wavelength. The x-axis here is the wavelength, and the y-axis is the attenuation length of the sample. A longer attenuation length means that light can penetrate deeper. And this green curve here shows the attenuation length of the mouse brain. Two-photon usually operates in the regime here. As we can see, when we shift longer and longer into the wavelength, we can encounter two sweet spots, one at around 1300 nanometers, and the other in around 1600 nanometers with the highest attenuation length. So it's a no-brainer that if we want to image the mouse brain and image the deepest, we'll use these two wavelengths. However, when we shift longer into the wavelengths, the photon energy will be lower. So instead of doing two-photon excitation, we'll do a three-photon excitation. Here is a comparison of two-photon imaging at 920 nanometers and three-photon imaging at 1320 nanometers. This is the imaging of a scattering beats phantom sample. As we can see, when we image deeper and deeper into this sample, even for two-photon, there are backgrounds that start to appear above the focus. And this explains why for two-photon imaging, we can only image up to 600 microns. Because even for two-photon, we can also see fluorescence above the focus that will contribute to the background that will degrade the image contrast. However, for three-photon, using a long wavelength, 1300 nanometers, you can see the focus is still really sharp in the focal plane when we are imaging deep into this sample. So now you may ask, can we just use two-photon with a longer wavelength? Can it help with the imaging depth? Well, the simple answer is no. Even using two-photon at a longer wavelength, as you can see in the example here, there are still backgrounds above the focus. So this background will also degrade the contrast of the image when we image deep into a scattering tissue. So now the logic is clear. We need the high nonlinearity of three-photon and also a long wavelength together to image deep in the scattering tissues like the mouse brain. Here is an example of imaging the mouse brain, and for the neuron-labeled brain, we can image to around 1 millimeter below the surface, and for blood vessel imaging, we can image 1.2 millimeter below the surface. And this is twice the imaging depth that 2-photon 920 nanometer imaging can do. So now we know how to image deep using a 3-photon high nonlinearity and a long wavelength 1300 nanometers. But how can we go wide? In order to do that, we need a large field of view microscope. So here I have my lab rat Aaron to introduce live in the lab our 3-photon large field of view microscope. This is the large field of view microscope. The footprint is around 3 feet wide by 3 feet tall, and the setup is relatively simple. So the laser will go through this hole here, and it goes through these 2 steering mirrors to steer the beam onto a scanner. After the scanner, there are 2 scan lenses. To conjugate the scanning onto the second scanner, we scan perpendicularly. After the second scanner, we have a scan lens and a tube lens to conjugate the scanning onto the back aperture of the objective. After the objective, we'll put a sample here for imaging. Question? Sure, go ahead, Aaron. The objective looks really large. How big is it? If you want to measure the dimension of the objective, the height is around 5 inches and the diameter will be around 3 inches. Thank you. Sure, you're welcome. And the sample will emit fluorescence. The fluorescence will be collected by the same objective, and it will be refracted by this dichroic mirror onto the collection system here. In the collection system, we will have 2 channels. One sits in here and one sits at the back. So it allows 2 fluorescence color imaging simultaneously. That's it for the large field of view microscope. Aaron? So with the large field of view microscope, we can image a field of view of 3.5 millimeter in diameter. This is an image of a micromirror showing we can image both in x-axis and y-axis of 3.5 millimeter. And what's more, the transmission of the microscope is really good as well. At the edge of the field of view, the 3-photon excitation fluorescence intensity will degrade to around 50% compared to at the center. It means that the transmission of 1300 nanometer at the edge of the field of view is around 80%. So with this, we can start to image the mouse. You can see this is an image of the mouse brain surface. You can see very clearly the midline of the mouse brain and also all the blood vessels that surrounds it. So in 2017, we have been imaging the mouse brain below the surface of 1 millimeter at a field of view of 200 micron by 200 micron. Now we can image with a field of view of more than 200 times more with 3.5 millimeter of diameters and with the same imaging depth of 1 millimeter below the brain surface. And here is a stack image with the magenta labeled the blood vessels and the grayscale with a cortex-wide neuron imaging of GCAM6. Each bright spot in the images shows a single neuron. To conclude, with 3-photon using a longer wavelength and high nonlinearity, it allows us to go deep. And with a large field of view microscope, it allows us to go wide. Combining these two, we can image deep and wide in a highly scattering tissue like the mouse brain. And yet, it is a challenge that was hard to solve.